So what I'm going to do today is to tell you about the work that we're doing in the area of drug addiction. A number of years ago, we and other groups came up with the notion that addiction can be viewed as a form of drug-induced neuroplasticity. The idea being is that addiction requires repeated exposure to a drug, and that because the lasting effects of this repeated drug exposure can last a lifetime, and an addict can remain at increased risk for relapse for a lifetime, we can hypothesize that changes in gene expression might be involved. So according to that idea, drugs of abuse, even though they interact initially with proteins at the extracellular aspect of the synapse, eventually signal inside of cells to the cell nucleus, where drug regulation of transcription factors, and as I'll show you, many other types of nuclear proteins, lead to stable changes in the expression of specific target genes, which then somehow summate to alter the function of nerve cells, the function of their circuits, and lead to the behavioral abnormalities which we define as an addiction syndrome. One of the interesting corollaries of this is that all current medications used to treat addiction focus on these extracellular mechanisms only, molecules that are receptor or transporter ligands and the like, leaving unexplored literally many thousands of proteins inside the cell that could potentially be drug targets, and I'll come back to that at the very end of the talk. In order to do this kind of research, we need to know where in the brain to look, and for that we've relied on a large literature over several decades that has defined so-called brain reward regions. This cartoon shows a sagittal section of a rat brain, a rodent brain. Human brain is highly homologous, identifying these brain reward regions. The focus is on dopaminergic neurons in the ventral midbrain, the ventral tegmental area, and the forebrain projections of these nerve cells to many limbic regions. I'll focus on the nucleus accumbens, but also very important are other regions like prefrontal cortex, hippocampus, amygdala, among many others. Now, historically, my lab focused on two transcription factors, deltafosb and Creb. And it's interesting to look back over the years and take stock on how we came across these two factors. They were candidate approaches, but at the same time, they both reflected a more open-ended approach. So for deltafosb, we discovered deltafosb in the context of addiction because it was the only phos family transcription factor that was induced after chronic drug exposure. And that time course is depicted in this cartoon, where all other phos family mRNAs and proteins are induced rapidly but transiently because the mRNAs and proteins are unstable. We found deltafosb as a minor constituent of an acute phos response in response to a single dose of drug. And the unique thing about deltafosb is that it can be stabilized through phosphorylation and other mechanisms, so that in response to repeated drug exposure, this small amount of deltafosb, quite a minor contribution acutely, begins to build, and after chronic drug exposure becomes the predominant phos-like transcription factor in the brain, in nucleus accumbens in particular. So the family of proteins encompassed by phos and its Jun partners are called AP1. Creb, the other transcription factor we focused on, shows a very different type of time course. And we came across Creb because it was a known effector of the cyclic AMP pathway. And we identified the cyclic AMP pathway very early on, over 30 years ago now, by doing what we would now call proteomic work, two-dimensional gel phosphoprotein electrophoresis, and identifying many substrates of protein kinase A being upregulated in the context of drug addiction. We've looked at Creb, found it too being upregulated, but with a much more typical time course compared with the unique time course of deltafosb. And over the ensuing couple of decades, we've accumulated a lot of data about how deltafosb and Creb work. Summarized in this slide, and I want to thank two former postdocs in my lab, Bill Carlazon at Harvard and David Self, who's at UT Southwestern, for contributing to most of this work. We know that in response to all drugs of abuse, deltafosb is induced selectively in D1 type medium spiny neurons. I'm going to define that in just a few moments. The one exception are opioids, which induce deltafosb both in D1 and D2 medium spiny neurons. When deltafosb is induced in the D1 type of medium spiny neuron, we have a lot of data that increases in animal sensitivity to drug reward, leading to an increase in drug self-administration as inferred craving and relapse through positive reinforcement. Creb, in many ways, does the opposite. It is activated in both major subtypes of medium spiny neuron within the nucleus accumbens and serves a classical negative feedback homeostatic role in mediating drug tolerance and dependence in reducing an animal's sensitivity to drug reward. It too increases drug self-administration and relapse, we presume, through negative reinforcement. Now, starting at about 10 years ago, when we moved to Mount Sinai, a little bit longer than that, maybe 15 years ago, we decided to take a step back from transcription factors and look more broadly at what else might be changing in the cell nucleus. And now this cartoon is summarizing what that encompasses. We know that a mammalian genome has about 3 billion nucleotides of DNA wrapped around octamers of histone proteins to form the unit of chromatin called a nucleosome. Nucleosomes are then organized in very standardized ways in the most condensed form, what looks like a chromosome. And it's remarkable that 3 billion nucleotides of DNA, when stretched out linearly, would be about 2 meters in length, can fit in a condensed form within a microscopic cell nucleus. And this organization allows that condensation. This is really important because the degree of condensation determines whether the spans of DNA are active or not, functionally active. Spans of DNA where nucleosomes are spread far apart can be active, can encode RNAs. DNA enclosed in more condensed forms of chromatin are inactive. And that means that we can use this understanding to help us identify drug-regulated genes. Most importantly, understand mechanisms underlying that regulation. Prior to the study of chromatin, studies of transcriptional mechanisms per se relied on in vitro data, even though we know what happens in cell culture, even cultured neurons, is not always a good reflection of what happens in the adult intact brain. And by analogy with mechanisms of development and cancer biology, where some types of mechanisms governing the opening and closing of chromatin, once they occur, are permanent, our hypothesis was that perhaps drugs of abuse induce similar semi-permanent changes in chromatin structure that drive some of the long-lasting features of drug addiction that are seen behaviorally. Now I'm going to show you a little more detail at this level of analysis. The DNA double helix are these gold stripes wrapped around octamers of histone proteins forming a nucleosome, showing the two broad ends of the spectrum of chromatin, active open, condensed closed, indicating the many types of modifications that control the transition of chromatin from one state to the other. Acetylation, phosphorylation, methylation of histones, methylation of the DNA itself, and so on. These are extremely complicated processes where these have been possible to explore in greater detail, like stem cells or yeast cells. Hundreds of proteins are recruited to the vicinity of a single gene to mediate its activation or its inactivation in response to an extracellular stimulus. The idea that we put forward in the area of addiction is that drug-regulated transcription factors like deltafosb and Creb, many others, would bind to regions of DNA where their specific response elements are relatively exposed and mediate some of these transitions to more open or more closed chromatin. The approach we took when we moved to Mount Sinai is to use next generation sequencing. So RNA sequencing to capture RNAs that are upregulated, another set that are downregulated in animal models of drug exposure. overlay on top of RNA sequencing, chip sequencing, and related measures to identify changes in chromatin that would reflect or even mediate, in some cases, those altered transcriptional states, chip sequencing for transcription factors, and so on. The idea being that looking at intersections in these Venn diagrams, that we would have much greater confidence in the genes identified than relying just on one platform, RNA-seq alone, chip-seq alone, and so on, each of which comes with a relatively high false positive and false negative discovery rate. And we have increasing evidence to justify that general experimental approach. So let me give you an example of how we recently used this idea. This is an experiment that Dina Walker led. The self-administration experiments were run by Erin Kalapari. Erin is now at Vanderbilt. Dina literally is on her way across the country now, taking on a job at Oregon. And what they did, along with others in the lab, was have mice either self-administer cocaine or saline volitionally for a period of time. A group of animals were harvested 24 hours after that self-administration period. Other animals were allowed to withdraw in their home cages for 30 days, at which point animals were given an acute injection of either cocaine or saline. So 6 experimental groups, and then we harvested 6 brain areas listed here, which reflect key regions in the brain's limbic circuitry. Now this 30-day and then we did RNA sequencing on all the tissues. This is a big RNA sequencing experiment, very expensive. This 30-day withdrawal period is important because in rodents, especially with some human data also, what we know is that an animal's craving increases during this period. That's shown at the inset toward the right, where if an animal is put back in a cage after 24 hours, they will return to a lever and press it. Even if the lever is turned off, it no longer delivers cocaine. It's inferred as craving, relapse. Where if you wait 30 days and repeat that experiment, the animal does so to a much greater extent. And that difference is considered incubation. So we're interested in this, what happens during this withdrawal period that increases an animal's interest in drugs of abuse. This is a lot of data. I'm just giving you one glimpse of the data in this slide. This is using machine learning, where we asked the computer to identify genes that were uniquely regulated at this 30-day time point. So what I'm showing you here for nucleus accumbens only, we have similar data for all the other brain areas. What are the genes that are upregulated in yellow or downregulated in blue? For those of you who don't know heat maps, each vertical line is an individual RNA. That are regulated uniquely at the 30-day time point, either in response to a saline exposure or in response to a cocaine exposure. One might call these withdrawal or incubated genes. One might call these primed or desensitized genes. And one can, and then in the panel to the right, looking at genes that are uniquely regulated only in response to that priming dose of cocaine. So you could see in this bar where my arrow is pointing, that shows the transcriptional response to an animal's first ever exposure to cocaine 1 hour later, 1 cocaine dose 1 hour later. You can see the dramatic reorganization of the nucleus accumbens transcriptional response to a cocaine challenge 1 hour later in animals that a month before had a history of cocaine self-administration. Now, we did these experiments on C57 inbred mice, but we know, and many other labs have shown, that the behavioral responses of inbred mice vary tremendously, not only in drug abuse models, but in stress models, eating models, and many others. And Dina took advantage of this using another machine learning approach to come up with what we call an addiction index, a major inference, but to get a sense, take advantage of the behavioral variability to identify those animals that look like they're more, that they show greater responses to cocaine than others. So now this heat map is not showing genes, it's showing different behaviors. Machine learning was used to come up with a set of behaviors that we thought reflected greater addiction-like behavioral responses as best as we can ascertain in a mouse. Animals that self-administered saline showed low levels of this addiction index by definition. Animals that self-administered cocaine showed a wide variability. Those with higher levels of addiction-like behavior, those with lower levels of addiction-like behavior, even every single one of these animals, even those at the low end of the spectrum, still learned to self-administer significant amounts of cocaine. And then we asked the question, how do the various individual differences in addiction-like behavior compare with transcriptional mapping? So in these heat maps, I'm now going back to showing you genes whose up or down regulation is either positively correlated with the addiction index in red or negatively correlated with the addiction index in gray under these acute, incubated, and primed cocaine conditions that I described earlier. I'm showing you the nucleus accumbens and ventral hippocampus as a counterpoint to make one particular interesting point, and that is that the genes that show particular transcriptional responses at the 30-day time point, incubated and primed, they look like most of the same genes were affected in response to an animal's first ever cocaine dose, although mostly in opposite directions, suggesting that one might be able to predict an animal's future addictiveness simply based on their transcriptional response to their first ever cocaine exposure, even though the direction of response of individual genes may vary. You could see that pattern quite apparent in nucleus accumbens is not evident in the ventral hippocampus. Now we can look into these data sets of genes that are significantly correlated negatively or positively with the addiction index and ask what are the predicted upstream transcription factors that might drive those genes. Those are listed here. Creb, we were very happy to see that Creb and AP1, meaning phos family transcription factors, were among the most highly inferred. That was reassuring. It meant that our earlier focus on those transcription factor families were wise guesses. But it also turned up transcription factors that have never before been studied in addiction models or barely studied in brain at all. And I'll come back to one family in particular, the E2F family, which we have since studied. I did want to say that in parallel, more recently, Caleb Brown and many others in the lab have now carried out an identical experiment looking at heroin self-administration. This gives you a bird's eye view of the data set of these chronic 30-day time points of an acute heroin response, this incubated heroin response 30 days after heroin self-administration, or primed heroin response 30 days after heroin self-administration following acute heroin challenge. And you can see transcriptional responses across the 6 limbic brain regions that were studied, and we're very interested in now diving into this data set further and comparing it to the cocaine responses as well. Now, everything that I've talked about thus far refers to differentially expressed genes. That these heat maps reflect. Yellow or blue, an RNA that's expressed at higher or lower levels under one condition versus another. But the magnitude of the data sets means that we have the capacity to use much more sophisticated bioinformatic approaches, such as various types of co-expression network analyses. And we've been working with Shansha Zhou and Bin Zhang at Mount Sinai with one particular approach, abbreviated MEGANA, which gives you the chance to break the genome into correlated modules or networks of genes that are co-regulated within our data sets. This gives you a view of the entire transcriptome of the nucleus accumbens under control or cocaine conditions. Each color is a different module. You can see a dramatic reorganization of the transcriptional structure by cocaine. You can then map these individual modules in what's called a Surcos plot shown to the right. Each slice of a pie is a different gene module. The genes in a module are collapsed, grouped simply on the basis of their correlation within our data sets, having nothing to do with their known biology. The one at 12 o'clock is the most significant, gets less significant as you go counterclockwise around the Surcos plot. You can ask what the major gene ontology terms are of genes within that assay, and one can use the concentric circles to ask whether differentially expressed genes are represented within those Surcos plots. So what I'm showing you here is now a very complex data set developed by Dina Walker that compared the effects of acute and chronic cocaine in males and females after a period of normal housing or stress during adolescence. And I'm gonna just highlight one finding that Dina had on the heat map at the very right, showing very broad differences in the effect of chronic cocaine in control females versus control males, again, focusing on nucleus accumbens, very broadly non-overlapping changes in genes, less than 10% overlap. But in mice that are stressed during adolescence and then exposed to chronic cocaine, you could see collapses these broad transcriptional differences. So there are a lot of bioinformatic tools that enable us to look at these vast data sets and begin to make sense of them and formulate hypotheses. Now, everything that I've talked about thus far has focused on bulk sequencing, bulk RNA sequencing on crude homogeneous of nucleus accumbens, even though we know that every brain area is comprised of many different cell types. For the nucleus accumbens, the major nerve cell type is called the medium spiny neuron, the major projection neuron of the brain area, divided into two main subtypes based on the type of dopamine receptor they express, D1 or D2. And these largely map anatomically onto the direct and indirect projection pathways. So most D1 containing medium spiny neurons mostly project back to the midbrain. They're the direct projection cells. A small subset are indirect projecting. Most of the D2 cells are indirect projecting, project to the ventral pallidum. And our lab and many other groups have contributed to an appreciation that these two nerve cells play different, in some cases opposite, roles in cocaine and other addictions. So in an earlier study, Mary Kay Lobo in my lab showed that activation of D1 medium spiny neurons promotes drug reward. Activation of D2 cells has the opposite effect. A few years later, Aaron Calipari, using fiber photometry, showed that acute and chronic cocaine, with many more complications, I'm grossly oversimplifying the data, activates D1 cells, but suppresses the activity of D2 cells. And I want to emphasize that these are just data from my lab. Many other labs have come up with similar research findings over the years, driving the importance of doing the type of sequencing studies on selected cell populations. So more recently, Philip Muse, a postdoc in the lab, has started to do this. He first used ATAC sequencing, which provides a means of looking at the spacing of nucleosomes within a group of isolated cells. Philip isolated D1 medium spiny neurons and D2 medium spiny neurons under the 4 cocaine conditions that I've already described: control, acute, withdrawal, 30 days withdrawal, 30 days withdrawal plus the priming dose, and now comparing the nucleosome spacing responses between the two cell types. So one sees very interesting comparisons. Under control conditions, you can see that in general, overlaying at the transcription start site genome-wide, many more genes are open in D2 neurons than they are in D1 neurons. This was a surprise, but it correlates with RNA sequencing that Hope Kronman in the lab did, that actually many more RNAs are indeed expressed in D2 medium spiny neurons at baseline than in D1 medium spiny neurons. But in response to cocaine, there are tremendous differences. The genome-wide differences in D2 cells are relatively modest, but dramatic differences are seen in D1 cells, with acute cocaine causing a dramatic activation of many genes, presumed activation, enhanced spacing at transcription start sites, and effects sustained despite 30 days of withdrawal, and in response to a priming dose of cocaine, is still further opening of the chromatin. This is looking at the identical data in a very similar way. Now, each horizontal line would be a given genomic region, centered again around the transcription start sites of genes, showing this dramatic opening of the chromatin and dramatic increase in the ATAC-seq signal in D1 cells, not so much in D2 cells. One can validate the data. For example, the gene encoding the D1 receptor is open in D1 medium-spiny neurons, not in D2 medium-spiny neurons. The opposite is true for the D2 receptor gene. And now one has a data set encompassing the entire genome. One can ask, well, what does the phosphate gene, just as one example, look like? You can see that the phosphate gene is relatively closed under control conditions. It shows signs of opening, but only in D1 cells, consistent with what I said, that it is induced only in D1 cells in response to acute cocaine. It looks like that opening is sustained during withdrawal, whereas little effect is seen in D2 cells. So you could just illustrating how these data sets can be utilized. More recently, Philip has now accumulated equivalent data by RNA sequencing of the same isolated cell types, now looking at the 3 time points versus the saline controls, looking separately at D1 medium-spiny neurons versus D2 medium-spiny neurons, and again, seeing dramatic priming of genes in terms of their now gene expression in the cocaine self-administration, 30-day withdrawal cocaine priming condition. One can look into these prime genes and ask what are predicted upstream transcription factors. Once again, the E2F family becomes highly identified as a top upstream regulator. And if one drills into what are these genes, we were very heartened to see that FosB is indeed one of the topmost prime genes in D1 medium spiny neurons, an effect not seen in the D2 cells. So again, going back historically, sometimes one's hunches are good, but it's great to see their validation through open-ended, unbiased data sets. Now, drilling down one step further, not only looking at individual cell types, but looking at single cell sequencing, I'm showing you on the top work done by Ann Schaefer's lab, where she has taken whole nucleus accumbens, done single cell sequencing of all cells in nucleus accumbens, and one can see the different cell types that one would expect to see in any brain tissue. But very interestingly, using standard single cell sequencing paradigms, the computer does not differentiate D1 and D2 medium spiny neurons. One can look at this broad group of cells colored green here and show which of them are expressing D1 receptors and which are expressing D2 receptors. And there's a broad distribution of both across this very similar population. This is very similar to what many other groups have published for striatum previously, and it suggests that medium spiny neurons may be very similar to one another from a molecular point of view, and then what differentiates the functional properties of D1 and D2 cells primarily may be their circuitry, the way they're connected, afferently and efferently, and not so much the constituent genes that are expressed. What Philip has done has gone one step further, he has taken FACS isolated D1 medium spiny neurons from nucleus accumbens and done single nuclear sequencing on just the D1 population. So just isolating the positive cells that are depicted here and expanding what heterogeneity can be observed. The computer identifies over 20 different clusters. Some of the clusters are interesting. One can identify a cluster that also expresses D2 cells. In fact, we know that about 10% of nucleus accumbens neurons do express both cell types. One can also express a cluster that's enriched in immediate early genes like phosphate and ARC and others. But this heterogeneity of D1 medium spiny neurons is hard to know what to do with. And we believe that single cell sequencing may not yet be ready for prime time for us to identify cocaine-induced transcriptional abnormalities until we do two things. First, better appreciate what this heterogeneity reflects, and number two, increase the depth of sequencing of single nuclei, because right now one can only identify a few thousand transcripts of the many tens of thousands of transcripts that we know are expressed within a given cell type. So that brings me to the interest that we have, and that it's very nice to show that 30 days of withdrawal from chronic cocaine or heroin self-administration causes this dramatic, long-lived, sustained reordering of the transcriptome of nucleus accumbens and other brain areas. But what is mediating those changes? What's responsible for the chromatin scars, as we call them, within the chromatin architecture around these genes that show abnormal levels of expression that mediate these effects? Now, in the old days, meaning up until a few years ago, the way we approached this was simply by guessing which chromatin modifications might be of interest. We carried out chip sequencing, work done by John Fung and analyzed by Li Shen and many others in our group over the years, looking at histone modifications that other labs have shown in other tissues and other systems being important. And we were disappointed by those data because the overlap that we got with cocaine-regulated transcriptional abnormalities was relatively small. So again, we decided to go back and take an unbiased approach. And I wanted to thank Ian Mays, but also Philip Muse, who have contacts with Ben Garcia's lab at Penn, and one of Ben's postdocs, Simone Sidoli, who's now has his own lab at Albert Einstein, and collaborating with them to take an open-ended proteomic approach, looking at all the hundreds of different types of histone modifications and other protein modifications within nuclei that can occur in the context of cocaine administration. So I'm showing you here data from Philip, again looking at acute, the 30-day withdrawal incubated condition, the 30-day withdrawal primed condition, and now I'm giving you just a high overview of some of the histone modifications that we've looked at. In red rectangles are those that are significantly differently regulated under the different conditions, and many of these are modifications that it would have taken us a long time to get to if we were going one modification at a time. These two types of modifications in particular. Philip's eyes were particularly drawn to H2AZ. H2AZ is a substitute of histone H2A, which can be added into certain nucleosomes in different forms of plasticity. And what he found is a marked enrichment of nucleosomes for total H2AZ or for H2AZ acetylation in acute cocaine conditions, and a marked depletion of H2AZ or H2AZ acetylation in these 30-day withdrawal conditions, just looking at nuclear extracts in total. So what Philip has started to do now is to perform chip sequencing on these different types of histone modifications, and I'll show you data for H2AZ. Now, organizing these plots around predicted enhancer regions, where we would expect, based on other data, changes in H2AZ occupancy to be particularly focused. I want to thank Li Shen and his group, Artie and Molly, for helping us do these enhancer analyses based on our own ChIP-seq data, using marks that have been identified by ENCODE as marking enhancers, but what we know are the maps for these regions within the nucleus accumbens tissue. you can see that that after an acute administration of cocaine, there is the dramatic enrichment of H2A, total H2AZ and H2AZ acetylation, going from control to acute, consistent with the proteomic data. And notice the almost complete depletion of these two marks in both the 30-day incubated and primed conditions. Really the most dramatic map that we've seen yet. And even more encouraging than that is that when we overlay these H2AZ chip sequencing data with our group of RNA-seq prime genes, we're seeing much greater overlap within these mechanisms as ever seen before by guessing. So again, underscoring the importance of unbiased, open-ended data, having gene discovery data, open-ended data guide one's hypotheses. So we're now drilling into these specific genes involved, doing RNA and chip sequencing on the other marks and proceeding accordingly. Now, everything that I've talked about thus far has focused on correlations. And I wanted to emphasize that my lab has invested a lot of effort over the years in also driving causality, using open-ended data sets to perform experiments to establish causal roles for specific genes. So I want to return to this family of genes called E2F, a family of transcription factors, barely studied in brain, barely studied in nerve cells prior to this point, certainly never implicated in nucleus accumbens or drug addiction. Yet this gene kept on coming up on our open-ended data sets as being highly regulated. So based on these inferences, Hannah Cates launched some studies of E2F that I wanted to highlight. So first experiment, in a study that Hannah did with Casey Larner. Hannah is a former graduate student. Casey's a graduate student who's about to defend this July. They first were able to show that cocaine indeed induces E2F family in the nucleus accumbens, an effect seen only in D1 medium spiny neurons, not in D2 medium spiny neurons. Let me just back up a point by making the comment that E2F3 is the only one of 7 E2F family members that's expressed appreciably in the nucleus accumbens, very low levels of expression elsewhere. The E2F3 gene encodes 2 RNAs, E2F3A, E2F3B by alternative splicing. E2F3A is the only transcript that's regulated by cocaine, and that regulation is specific to D1 medium spiny neurons. Citing an earlier study done by, published by Hannah Cates a couple of years ago, using viral vectors that overexpress or knock down these various types of E2F alternative splice variants within all nucleus accumbens neurons, and looking at their behavioral consequences in a condition-place paradigm on this left panel. So using a low dose of cocaine, where a control animal shows very minimal responses to cocaine under control conditions, in animals that are overexpressing E2F3A in the nucleus accumbens neurons, there's a robust rewarding response to that low dose of cocaine, no effect when E2F3B is overexpressed. The converse is true using a microRNA that and a higher dose of cocaine that produces a significant rewarding signal under control conditions. Knocking down endogenous E2F3A blunts that rewarding response. Knocking down E2F3B has no effect. More recently, Fredison Martinez Rivera, using the same microRNA, can show that this knockdown also suppresses measures of cocaine self-administration, and more recently, using viruses that overexpress selectively in D1 or D2 type medium spiny neurons. These are Cre-dependent vectors using D1 or D2 Cre mice. Fredison was able to show that the ability of E2F3A overexpression to enhance cocaine rewarding responses is seen only upon overexpressing E2F3A in D1 medium spiny neurons, no effect in D2 medium spiny neurons. One can then go on and look at what's happening transcriptionally. This is going back to the earlier paper by Hannah, now asking what effect does E2F3A overexpression have on the transcriptome of nucleus accumbens of mice? And how do those effects compare to the actions of cocaine? One can see in this heat map lots of similarities. Many of the genes that are up or down regulated by cocaine in nucleus accumbens, the same genes are up or down regulated by E2F3A overexpression. One can look at that statistically using a bioinformatic test called RRHO, rank-rank hypergeometric overlay, that simply provides a way to look at whether two conditions produce the same effect along this axis that I'm showing you with my cursor, or opposite directions in the with a different type of slope shown here. This is looking at overall transcription. Hannah was able to also look at whether E2F3A replicates the effects of cocaine on alternative splicing. This cartoon at the upper right shows 4 different types of alternative splicing. I'm not going to go into the different types of splicing. They're depicted here by heat maps, simply showing that in each case, overexpression of E2F3A recapitulates a portion, a subset of the splicing changes induced by cocaine. So this is a transcription factor never before looked at in cocaine models, never before looked at in neurons or brain, based on our open-ended data pointing toward E2F3A. Following those bioinformatic leads, we're able to discover a new biology underlying cocaine action. In the last 5 minutes of my talk, what I'm going to do is to shift gears a little bit and bring us back to deltafosb. The first experiment I want to show you is to connect E2F3A to deltafosb in a study that Hannah and Casey published last year in eNeuro. What they did was to provide a series of lines of evidence suggesting that, in fact, E2F3A induction is one of the drivers of deltafosb induction, not surprisingly perhaps, since cocaine induces both within the same cell type. And by the way, unpublished data suggests that the opposite is not the case. Deltafosb does not influence E2F3 expression. First, we can show that cocaine administration induces binding of endogenous E2F3 to a region of the phosphate promoter containing a predicted E2F site. When we overexpress E2F3 virally, E2F3 is recruited to that same site. And we can show that when we overexpress E2F3A but not E2F3B, we can induce phosphate expression in nucleus accumbens. And we can show the opposite when animals are treated with cocaine to induce phosphate. Knocking down E2F3A blunts that response. Now, the kind of data that I'm showing you in this slide represent the state of the field. This is what people do. They overexpress E2F3A. They knock down E2F3A. We have overexpressed phosphate, knocked out phosphate. We've manipulated other chromatin modifying factors. But when we manipulate any one of these genes, we're affecting total cellular levels. We're affecting total cellular levels of E2F3A, and it's binding not only to phosphate, but probably thousands of other sites in the genome. So the question is, are there better ways to show better quality of proof to establish a role for a given gene in mediating a phenomenon? And I wanted to return to earlier data by Vincent Vialou in the lab, implicating Creb as actually also upstream of cocaine's induction of deltafosb. Turns out phosphate inductions requires 3 transcription factors, E2F3A, Creb, and SRF. So this is now a study done by Liz Heller. Liz is at Penn now, published several years ago, where she took the FOSB gene because we knew a lot about it. We wanted to ask whether chromatin modifications at the FOSB gene only can be seen as causally involved in controlling FOSB induction. So the question is the following. Can we manipulate in the mouse, in nucleus accumbens neurons only, one type of histone modification, and only do that modification at the FOSB gene? We're not overexpressing or knocking down a histone modifying enzyme, which would affect the effects of that enzyme at hundreds, thousands of sites. We only want to manipulate it at FOSB. So what Liz did was to take a series of zinc finger proteins, synthetic transcription factors, artificial proteins, couple them to a G9A, a gene that produces a repressive histone methylation mark, or to P65, a gene that indirectly adds acetyl groups to histones. And she found one particular zinc finger that bound specifically to the FOSB site. When it was fused to this histone methyltransferase, she suppressed FOSB expression. When she fused it to this protein that increased histone acetylation, she increased FOSB expression. Those effects are shown here. We were able to show that these effects are specific to methylation, acetylation, methylation, not to other modifications. And importantly, the zinc finger protein that worked in vivo did not necessarily work in cell culture. So Liz had to do a screening method of 20 zinc finger proteins by itself, fused to the methyltransferase, fused to the acetylating moiety. So 60 viral constructs in series screened in vivo, an enormous amount of work. Brute force is sometimes one of the best features that we have in our arsenal. Liz was able to provide indirect evidence that these effects are quite specific to the FOSB gene. This is showing FOSB and deltafosb transcription, and showing these predicted behavioral effects. That when we treated animals with a higher dose of cocaine that increased cocaine locomotor activity, repressed FOSB expression, we repressed that behavioral response. A lower dose of cocaine having no effect under normal conditions, now activating FOSB through this acetylating mark, producing an increase in behavioral responses. So one step further, however, is what about Creb? Can we bring a transcription factor to the FOSB gene and study what happens? The answer is yes. This is work done by Casey Lardner and Peter Hamilton. What they did, building on a method that was in Jack Lorsch's recent publication last fall. They used CRISPR. CRISPR offers a way to target a dCas9 protein to a specific gene using a complementary guide RNA. They fused Creb to the dCas9. The idea is that this would be a way to bring this protein complex, including Creb, to a gene of interest. Using a lot of controls, they were able to obtain evidence that when they overexpressed a constitutively active form of Creb, one phosphamimetic mutation of Creb, along with a guide RNA targeting the FOSB gene, they showed induction of FOSB. A single point mutation of Creb that kills it makes it constitutively inactive. A dominant negative mutant obliterates that activity. So what Casey Lardner has done for her thesis, this is a advertisement for what you'll hear from Casey in July, now using RNA sequencing from tissue taken from animals, where Casey is overexpressing these CRISPR constructs selectively in D1 medium spiny neurons, in D2 medium spiny neurons, or in both cell types. She can then do RNA sequencing and ask to what extent does this deltafosb induction, now inducing the endogenous FOSB gene, replicate the effects of cocaine self-administration. And you can see a significant replication of cocaine's effects when FOSB is induced endogenously in D1 medium spiny neurons, but not in these other conditions. So we think that these CRISPR constructs represent a major step forward in enhancing the quality of proof that we have in our experiments, because we're inducing endogenous genes much more physiologically than overexpressing or knocking out a gene, which is manipulating genes to a magnitude that's just not seen under natural or pathological conditions. So I realized that what I've been talking to you about today is a gross reductionistic approach from something involving the brain and behavior, looking within nuclei at transcriptional and epigenetic abnormalities induced by drug exposure. We think that the transcriptional and epigenetic maps that we're accumulating provide a template, a periodic table, so to speak, of many types of proteins at the synapse, at other subcellular compartments that are mediating the functional aspects of addiction, which now can represent drug targets. But I also want to highlight the other interesting idea that perhaps some of these epigenetic mechanisms might be drug targets themselves. And Yasmin Hurd's lab has done some beautiful work targeting epigenetic mechanisms directly, since many of these mechanisms are now direct targets for cancer chemotherapeutic agents. Doing clinical studies with them would be relatively straightforward. So with that, I wanted to conclude by making just a few comments. I hope I've convinced you in the importance of open-ended investigation in a hypothesis-generating way to understand the ways in which the transcriptional organization of a brain region, and even a specific cell type in a brain region, is completely reworked in a drug addiction model. I focus today solely on cocaine, solely on nucleus accumbens, but emphasizing the importance of looking much more broadly within the brain's limbic circuitry, looking at other drugs of abuse. We are most interested in understanding the nature of these chromatin scars. I've given you some ideas of how we're going about identifying these scars, again, utilizing open-ended chromatin biology assays. And the hope is that this will lead us to advance in therapeutics. So with that, I'm going to end my show, stop my screen sharing, if I could figure out how to do that, and take any questions that might come up. So if you guys type your questions into me through the chat function, I'll look at them, or if Veronica unmutes you, you can just ask me directly. Thank you.
Thanks so much, Eric. That was fantastic.
Hey, Allison.
Hi. So I just wonder whether you have any data looking at other drugs of abuse, and whether you think that this is the specific patterns associated with cocaine, or whether this is a generalized response to addictive substances, or even, you know, it doesn't even have to be a substance. I guess there are addictive behaviors as well. So how specific do you think this is to cocaine, or is it going to be a route into, and I saw on your last slide about pan-addiction therapeutics. Do you think that this, do you have evidence from your animal models at all that they're the same transcription factors and programs are operating with different substances?
Yeah, so we know that there are both. So when we look at our data sets thus far, or look in the literature, there are a lot of examples of pan-addiction effects. Deltafosb actually represents one of the best established pan-addiction mechanisms induced by all drugs of abuse. But there are also drug-specific effects, and we're very interested in looking at these genome-wide data sets to give us insight into these distinctions. I think both avenues will be very...
What about, I was thinking specifically actually about your new, the E2F3A finding, whether that...
Yeah, so we don't know the answer to that yet. So that will be one of the first things we'll look at in a heroin data set that we have now, to see whether it's implicated as well. We don't yet know.
Okay.
Mark Baxter asked a question. Vanna, go ahead. And then, you know, I'm cognizant of the time. I don't want to hog people's time. So Vanna, make your question really fast. I'll answer it, and then answer Mark's. And then we'll
really fast. now you have all these gene lists, and maybe you can use bioinformatics to predict if any existing drugs that we haven't thought about could be used for the treatment of cocaine or pan-drug addiction.
Yes, absolutely. That's another approach. And actually, Eric Shad's group and Bin Zhang and others here at Sinai have led the field and using some of these reverse translation type approaches. And we're very interested in looking at those data sets. Let me quickly answer Mark's question. It's, can I say something about the tension between hypothesis-driven versus hypothesis-generating approaches in the context of grant proposals, which is really important. As you got from my talk, I am much more keen on hypothesis-generating experiments than hypothesis-driven proposals. However, all of my grant proposals are hypothesis-driven because this is what NIH wants to see in R01s. Except for U grants or program grants, center grants, where the institutes are interested specifically in hypothesis generation, I start with these open-ended data sets as a justification for the hypotheses that I'm proposing to study. And in fact, I think having the open-ended start is a way to really strengthen a grant proposal because it's showing I'm not just picking a candidate gene out of thin air. I pick my candidate gene for a hypothesis-driven proposal based on open-ended data. So I think in some ways merging the two approaches gives you the strongest effect. Listen, guys, it's 2:01. I don't want to hog time. I'm available by email. Anybody has questions, send me an email. We had a great show today. We now have this FBI Cyber Series every Thursday at 1 o'clock. Paul Kenny's up next. So let's show him the same great turnout as we had this week, and we'll keep this going for the rest of the spring. Thanks, everyone, and stay safe, and look forward to seeing everybody in person at the other side of the pandemic. Ciao.